probably figured out that I love almost all of biochemistry, except for the math. But what I really love is translation, the process of making a protein based on the instructions of messenger RNA. This has been a recurring theme all the way from grad school to now when I want to do my own research. I love translation, so I want to tell you all about it. Kind of like a beautiful molecular dance that's taking place inside of a giant complex of proteins and RNA called the ribosome. And this dance is so beautiful and it's really, really accurate. It only makes like one mistake in 10,000 or so amino acids. And it does so because it's paying the cost to make sure that only the right tRNAs bring the right amino acids to be added, and that only the right amino acids are actually added. And so these amino acids are going to be the individual building blocks of the protein that the ribosome is piecing together based on the instructions in the messenger RNA. And the ribosome has these three main sites, these kind of rooms, the E, the P, and the A. So the P is the peptidyl site. That's where the growing chain is going to be held. But when a new tRNA comes and brings the next amino acid in the A site, basically the arrival site or the aminoacyl site, this is where that aminoacyl tRNA is going to come in, and it's going to be holding that amino acid that needs to be added next. The ribosome is going to kind of help egg it on so that it attacks the growing chain, and this way the growing chain gets transferred to that incoming tRNA. And now it's going to be a really awkward shape, and so with the help of some elongation factors and some GTP, it's going to kind of like shift over. And when it shifts over, well, now it's going to push that what was holding the chain into the E site. And this E site, this like exit site, basically what's going to happen here is that tRNA is going to fall out. And now your chain is in the P site again, and you have an empty A site. And this empty A site is where the next tRNA is going to come, and it's going to come in holding the next amino acid. And it's going to come in, like all of these are coming in with the help of an elongation factor. It's kind of like a chaperone. It's going to help make sure that the right tRNA is being brought and that it's like actually the right one before it's going to spend some GTP and leave. So you have GTP being paid in the cost of bringing the new amino acid, waiting to make sure that it's actually the right one, and then it'll spend that energy. Now you have to spend more energy in another elongation factor in order to push things over into the P site, and you're going to be spending energy when you're actually loading that tRNA, so when you're actually putting the right amino acid onto the tRNA. And so thanks to all of these things, your ribosome doesn't have to worry about actually having proofreading activity. So it can't like erase, it can't go backwards. Like your DNA polymerase and your things like this that are making copies of DNA, they can proofread, and so they can actually like go backwards and fix things. Your protein making machinery can't do this. It's not as big of a deal though if you make a mistake in making a protein versus making a mistake in your DNA, because the protein is just a single copy, and so if you make a mistake in one copy, it's not like it's going to influence things really that much. But if you make a mistake in your DNA, well, now you can pass on a mutation. And so, but the ribosome still wants to be making good proteins, right? And so in this way, we're spending a lot of energy in order to piece together these amino acids to make a protein. And so the whole process is really, really cool. And so the ribosome is going to go along. It's going to keep adding together all of these amino acids. It's going to get to what we call a stop codon, basically a signal that's going to tell the ribosome to stop making that protein. And so it's going to stop making that protein. And what's going to happen is instead of a new tRNA coming in with the next amino acid to be added, you're going to get a release factor come in. And the release factor, what it's going to do is it's going to, instead of egging on the tRNA to attack, or the new amino acid that was attached to the tRNA to attack, is going to kind of like egg on water to attack. And so water is going to attack this growing chain. And when you get water attacking that growing chain, the growing chain is going to be released. And so you get this released, and then you have all these other factors come in and kind of help the ribosome like come off the mRNA, and so it can be used again for new messenger RNAs and things like this. But basically, translation is this really, really awesome process. And so I want to tell you about it in a little more detail. So I'm not going to go into much detail in terms of the background and the messenger RNA. I have lots more posts on that, and I want to dive more and focus on the process of translation. But it's important to realize that what the ribosome is doing, it's doing based on a messenger RNA template.
And this messenger RNA template is going to be an edited RNA copy of a DNA gene. And so this messenger RNA is going to have the instructions for which amino acids to add. And then a tRNA or transfer RNA is going to bring the corresponding amino acid. The messenger RNA can be read in these three-letter words called codons. And basically, the tRNA with the matching amino acid is going to come and it's going to have a complementary anticodon. So tRNA is, like the name suggests, it's an RNA. And so it kind of has these three RNA letters, these three nucleotides sticking out that are going to match or complement the messenger RNA. And this, in this way, is going to help the ribosome be able to add the right amino acid, assuming that the tRNA is charged correctly. And so we're actually going to see how we spend money to charge the tRNA, because ribosomes, as much as I love them and I really, really love them, they're not that smart. They're only going to be able to tell if the codon and anticodon are complementary. They're not going to be able to tell whether the right amino acid was actually attached to that tRNA. But assuming that all goes okay, the ribosome is then going to be able to add these amino acids in the correct order. And then they're going to be able to, as the ribosome is piecing them together, they're going to be kind of pushing the older ones out of the chimney. And then those are going to fold up based on the properties of those amino acids to give you a pretty protein. So in a little more detail, let's look at how this is actually happening in the ribosome. And so first, let's look at ribosomes themselves. So here's an example of ribosomes. This is actually going to be drawn in backwards from how I'm showing this. So this is showing the ribosome has these three sites, the E, the P, and the A that we'll get into. And this is showing it in kind of the APE order. But ribosomes are made up of this large subunit and a small subunit. And so the large ribosomal subunit, or the LSU, it's going to have the peptidyltransferase center. So that's where we're actually going to get the pass off from the nascent chain, so from the growing chain in the P site to the incoming aminoacyl tRNA, so to the A site tRNA. This is going to be happening in the PTC, the peptidyltransferase center, which is in the large subunit. And then the small subunit, this is where you're actually going to have the tRNA binding to the mRNA. And we call the decoding center, which is where it's like the check place, like does the codon match the anticodon? So that's going to be in the small ribosomal subunit. And so you need both of these together in order to form a functional ribosome. So note that in prokaryotes, so like bacteria and archaea, your full ribosome is going to be your 70S, and it's going to have a 50S LSU and a 30S SSU, or yeah. And basically, the S refers to these things called Feldberg units, which relates indirectly to the size. It's basically based on the sedimentation rate, which is if you spin these things down, how fast do they sink in like a gradient? And so it's not directly additive, which is why 50 plus 30 might equal 80, but 50S plus 30S equals 70S. If you're talking about 80S, well, now you're talking about a eukaryotic ribosome, so like plants, animals, and fungi. And these are made up of a 60S LSU and a 40S SSU. So just if you come across these terms, that's what they mean. And basically, each of these is going to be made up of a bunch of proteins and RNAs. And so although we often think about enzymes or reaction helpers as being proteins, they can also be RNA. And in this case, they're RNA protein complexes. And the bulk of the hard work is actually being carried out by the RNA. So within this ribosome, you're going to have these three sites, the E, the P, and the A. The P site, the peptidyl site, this is where their growing chain is going to be held. The A site, this is like aminoacyl site, or I like to think of it as kind of like the arrival site. This is where the incoming tRNA is going to come with the anticodon that corresponds to the codon that is now in this A site. And so the A site codon is going to be the one that's going to be kind of like being read when the new tRNA comes in. You're going to get the pass off, as we'll see, and then we have the P one is going to get kicked into the E site, this like exit site, where it's going to then be able to like fall out. And so you have the E, the P, and the A site. Most of the time, the tRNA is going to be coming in the A site. The only exception is when you have the initiation. So in order to get things started, you have to have one of them has to be first. And that first one's going to, therefore, your peptide is only going to be one, and it's going to be in the P site, though. And so everything's going to kind of have to form around that initiator tRNA in the P site. When I talk about this initiation, basically it's important that the ribosome knows where to start and that it starts at the right place. Because if it starts out of frame, basically those codons are non-overlapping. So if it starts in the wrong place, you can kind of get all the letters be off. To make things even harder for the ribosome, although there is a start codon, there is a codon that tells the ribosome this is where to start. It only does that sometimes. And so the start codon is AUG, which is actually also the codon for methionine. So it's a codon for one of the amino acids. And so if the ribosome is already in the middle of like making a protein and it comes across an AUG, it's just gonna put in a methionine as usual. But this codon can also be a methionine and a start codon, meaning that yes, the ribosome does put in a methionine, but it also starts a new chain at that site. And so how does the ribosome know where to start? It gets signals from the part of the messenger RNA that's actually not part of the codon coding region. So it gets signals from the part like upstream or like before the actual start codon. So messenger RNA, this mRNA, is basically an edited version of the gene. And part of the editing involves removing regulatory information. So these like intron regions that are in between or like interrupting the exons, the parts with the actual protein making instructions. And so you have a part of the messenger RNA that has all the instructions, all those codons that are going to tell the ribosome add this, and this. But then you also, at the ends, you have these untranslated regions, these UTRs. So you have the 5' UTR and the 3' UTR. And the 5' UTR, this like kind of beginning sequence, this like front matter, it's going to have information that's going to help the ribosome know where to start. You also have help from, at least in the case of eukaryotes, we have this like a cap on the 5' end, which is going to bind to various initiation factors and help the complex get formed, help the initiation complex get formed. And then you have various RNA sequences and things like this that are going to help the ribosome know where to start. But you can still also get like alternative start sites and things like this, which can give you different proteins from the same gene. So it's kind of complicated and people are still trying to work out all of the details. Somehow, these initiation factors are going to help the ribosome get situated in the right spot where you have that initiator tRNA, which remember is also going to hold a methionine. It's going to be in the P site. In bacteria, they actually use like a special methionine that in the initiation, it's going to be kind of like a modified version. The whole ribosome doesn't get assembled until the small subunit kind of finds the right place. So the small ribosomal subunit is kind of going to get recruited by those initiation factors that the signals and the cap and things like this help recruit. And then what's going to happen is once the small subunit finds the right place, you're going to get the large subunit brought in and you're going to get everything assembled. And so now we have everything assembled, you can get that full ribosome and you can get translation happening. So remember the decoding center where the tRNA needs to match the messenger RNA, that's in the small subunit. So you're able to kind of find that start site and then bring in the large subunit, which has the peptidyltransferase center. And so it's actually going to be able to do that transfer of the chain. Before we even get this translation going, we're going to be spending energy. So anytime you see like GTP or ATP, this is going to tell you that you're spending energy. And so what I mean by spending energy, we often talk about ATP as being like the universal cellular currency, but for translation, we actually use GTP. In both of these cases, what we're talking about is breaking what we call high energy bonds. They have high chemical potential energy. Just like the ball at the top of the hill has high like kinetic potential energy, chemical bonds have high chemical potential energy and in some cases. And basically, the more uncomfy a bond is, the more potential energy it has. If you think of these phosphate groups, in a triphosphate, you have these 3 negatively charged groups that are held closely together. Negative charges repel one another, and so this is going to be really uncomfy, kind of like a clamped spring. Now, if you were to break it, kind of like unclamping the spring, well, now you're going to be relieving some of that uncomfiness. When you relieve uncomfiness, you release energy. So kind of like if you had a ball at the end of your spring and you release the clamp spring, the ball would fly across the room. If you have a triphosphate bond and you break that bond, now you release energy that you can use to do things like get molecules to join together in ways that might not be energetically favorable and things like this. We often use ATP, but in translation, you actually see GTP be used a lot. And so we'll get more into this later. But GTP is just like ATP, except this part is different. But the phosphate groups are the same, and so you get that same energy boost when you break one of them. And we're going to use a lot of energy to make sure that we have the right amino acids being added. Overall, we're going to see that we kind of spend about 4 triphosphates per peptide added. As well as we're going to spend energy for that initiation phase and when we're actually recycling the ribosome and things like this. So we'll get back to this. For now, let's go and look back at what happens once we have things assembled. Once we've done that initiation step and we have things assembled with the tRNA, with the initiator tRNA in the P site. So in this case, you wouldn't have the chain here, you would just have a single amino acid. But similarly to what happens when you're making the chain, you're going to have an empty A site. When you have an empty A site, you're going to have a codon exposed in that A site, and that codon is going to then be open to pair with a tRNA that has a matching anticodon. And so you have all these tRNAs floating around, but only the ones with the matching anticodon are actually going to be able to like stick if they float in there. And so if they float in there and they stick, what's going to happen is that the ribosome is going to be able to catalyze the transfer, so get it to be added to the chain. But the tRNA doesn't come alone. It comes with the help of an elongation factor, which is going to help make sure that it's actually the right one. It's not just sticking in there accidentally. And so these elongation factors, in the case of bacteria, it's F2. In the case of humans, it's EEF1A. So E for like eukaryotic elongation factor. And so basically what it's going to do is it's going to make sure that the tRNA and codon and anticodon match. This is really important because that's the only way that the ribosome knows what to add. And so we'll talk later about how it's important that the tRNA be loaded correctly, but it's also important that the right tRNA, that at least the anticodon-codon part matches. And so this elongation factor that kind of helped bring in that tRNA, it's going to stick around until it makes sure it's the right thing. And then it's going to spend GTP. So it's going to break one of those GTP bonds to form GDP and then fall off. Basically, this elongation factor, when it does that breakage, the energy from that is used to kind of lead to a conformational change or kind of like a shape shift in this elongation factor. And now the elongation factor goes from liking to bind to the ribosome to not liking to bind. And so once it doesn't like to bind anymore, it's going to fall off, and then it can be regenerated. It can get a GTP swapped back in, and then be used to bring in another tRNA. And so in this way, you're spending energy to make sure that the right tRNA gets added. Now what's going to happen, though, is that aminoacyl tRNA is in the A site, but it needs to get added to the actual chain. And so now we need to have an actual peptide bond form. And this peptide bond is going to form when the aminoacyl tRNAs, the amino acid part of it, is actually going to attack the growing chain. So if you see in isolation, you might see like a peptide bond formation drawn something like this, where we draw it as like this condensation reaction, where basically you have the carboxyl group of one amino acid link up to the amino group of another amino acid, with the loss of water. When you look at peptide bond formation, you might see something like this, where you get this condensation reaction between the carboxyl group of one amino acid and the amino group of the other amino acid. You have the alcohol and the hydrogen from this amino group joined together to leave as water, and voila, you've got a peptide bond. Although you could make this happen in vitro, this isn't how it actually happens in vivo for a couple of different reasons. One being that water wouldn't be a very good leaving group. The other is that we need to have these specified added together in the right order. We can't control like having exactly what amino acid flows into the active site, but we can dictate it with our tRNAs. And if we attach these amino acids to tRNAs, then we can join them together once they're already attached to the tRNAs. So what we'll see is that one of our amino acids is going to be attached to a tRNA in our P site, and then the incoming one is going to be attached to a tRNA, and then this incoming amino acid is actually going to attack the tRNA attached amino acid, the chain in the P site. So what we're going to get is that we have our incoming amino acid. So this is coming into the A site. The amino group from it is going to attack the carbonyl carbon of the peptidyl chain that's in the P site. So this growing peptide chain, our incoming amino acid is going to attack it. And this chain is going to be held on the tRNA. Now your incoming chain is going to get attached to that incoming tRNA. Now, how it's going to happen is through a nucleophilic attack. So we're going to have that amino group of the incoming amino acid is going to act as our nucleophile, and it's going to attack our electrophile. In this case, our electrophile is going to be the carbonyl carbon that's attached to this tRNA in our P site. What's going to happen is that our tRNA is then going to get released. How do these amino acids actually get onto the tRNA? Well, that's a couple-step process, and it's done by these aminoacyl tRNA synthetases, or these aa-RSs. These spend energy to make sure that you're adding the right amino acid to the corresponding tRNA, so that when the tRNA gets to the ribosome, when the ribosome doesn't know what amino acid is on there, you'll still have the right amino acid be added. These tRNA synthetases work in two steps. The first is that they need to activate the amino acid to get added to the tRNA. If we want to think about linking this amino acid to this tRNA, even if we deprotonated this hydroxyl group on our kind of empty tRNA, it wouldn't be strong enough to push out this OH minus, which would be a poor leaving group. A good leaving group would be something like an AMP molecule, something like adenosine monophosphate. This would be happy to go. This would be a much better leaving group. So we start by spending some of our energy. We use ATP in order to activate our amino acid. So we adenylate it. We add this adenosine monophosphate onto our amino acid. And now our carbonyl carbon of this amino acid is going to be next to this phosphate group. If we attack this carbonyl carbon, we can kick out this AMP, and this is going to be stable on its own. It's going to be much happier. You can think about if you were to kick off this OH minus, well, now it would be a strong base that's just going to come and kind of attack right back. But this AMP isn't going to attack back. Instead, it'll be happy on its own when we kick it out. So how are we going to kick it out? Well, we want our tRNA to kick it out. So now our tRNA, this is going to be a strong enough nucleophile to then attack this carbonyl carbon and kick off the AMP. And voila, we've got our amino acid attached to the tRNA. Now, when we go and we bring this tRNA into the active site, into our A site, we have this amino group that can then go and it can attack the carbonyl carbon that's held in the tRNA in the P site that has the growing chain. Now what's going to happen is when this nucleophile, our nitrogen nucleophile, is going to attack this carbonyl carbon, and it's going to then kick off this tRNA. So this tRNA is going to be our leaving group. So we are losing the equivalent of a water, but it's not happening in a simple one-step reaction like this. Instead, we're kicking off this We're kind of kicking off things in a couple different parts. This oxygen of the tRNA is going to be kind of constant there, and it's the carbonyl carbons that are going to be getting attacked. So things are going to be kind of kicked off. This bond here is going to be broken, and the tRNA is going to then have that O that can then get attached to the next amino acid to be added. These tRNA synthetases are cool, and they have a bunch of different tricks to kind of recognize the right tRNA and recognize the right amino acid. And I'm not going to go into that, but it's going to happen. It takes energy. The energy is worth it because your ribosome's not going to know what the heck the amino acid is when you go and you add it to the growing chain. It's just going to know if the anticodon matches the codon. So you want to make sure that it's the right thing. So it's going to be something like this. The new amino acid comes in the A site, chain gets transferred, and now what? You're stuck in the A site, but we need things to be in the P site. This is going to be a really awkward situation for the ribosome because, well, the codon anticodon is kind of getting the tRNA stuck in that A site. So it's kind of like held there. But the peptide exit tunnel is coming out from like the P site. So you kind of have your chimney of the ribosome over here. And so the chain was still going through that chimney, but now its base is shifted and it's all kind of rotated and awkward. And so you're going to get another elongation factor come in and kind of help push it along. And so basically this is another elongation factor. So the EEF2 in humans or EFG in bacteria, it's going to come and it's going to spend energy to push things over. And basically, so now you get things pushed. You have the old one pushed into that E site where it can fall off. And now your chain is held in the P site again. And you have an empty A site, which can now serve as the entry point for another tRNA. And so it's kind of like that song, like the monkeys in the bed, three little monkeys sitting in the bed. One came in and another one said, roll over, and they all rolled over and one fell out. That song is kind of like that, but it's happening at this level where the incoming amino acid attacks the growing chain. You get the transfer. Now things have to get pushed over. What was in the P site is now in the E site where it can go away. A new tRNA comes into the A site, and you get this going on and going on and going on. But eventually the ribosome is going to need to stop. And so it knows how to stop because there's going to be a stop codon showing up. So this is going to be UAA, UAG, or UGA. So sometimes you might see a codon table written in the form with the DNA. So it would be like TAA, TAG, or TGA. So that's a sequence that you would see in the DNA form. But in RNA, the T's are actually U's. So we have a uracil instead of thymidine in RNA. And so that's why we see UAA, UAG, and UGA. And this is what the ribosome is going to see. And when the ribosome sees one of those, so when one of those shows up in the A site, what's going to happen is that there's no tRNA that actually matches that anticodon. But there is this release factor that looks like a tRNA, and it can bind there and kind of like sneak in and therefore cause the release because it's not holding an amino acid, but it have the potential to kind of like activate a water in order to attack that chain. And so we get another type of substitution reaction, but here it's coming from the water and not from the incoming aminoacyl tRNA. And so what's going to happen is that you get the chain cut off and the peptide chain is going to be released. And so overall, you get this process like this. This process is going to be really, really powerful and really efficient and really, really accurate. And a lot of this accuracy comes because you're paying money up front to make sure that the right tRNA is loaded with the right amino acid. And this happens thanks to aminoacyl tRNA synthetases, or these aa-RSs. So that's one of the places that you'll see energy being spent. And because you're going from ATP to AMP, it's kind of like you were spending 2 ATP because you're going to have to spend an ATP in order to regenerate the ADP in order to regenerate the ATP. And so it's kind of like you're costing 2 there. And then we're going to spend a GTP to make sure that the right tRNA gets brought in. So that was that initial elongation factors. That would be like our F2 or our EF1A. And then we have another GTP to get things to scooch over in that ribosome. So where you're going from pushing that growing chain from the A site back to the P site where it belongs. So you free up that A site. And that was going to cost another GTP. So that was another elongation factor. So that was like our EFG. In order to recycle these elongation factors and all these helper factors, basically, these are what we call G proteins. So you might have heard of like signaling where you have G protein coupled receptors. Basically, a G protein is one of these little proteins and it has GTPase activity. It can break GTP and make GDP, but it can't make GTP from GDP. So it can't really remake it, at least not under physiological conditions. And so instead of trying to get this enzyme to remake the GTP to regenerate it, you actually have the GDP, get it drop the GDP and exchange it for a new GTP. So there are these exchange factors that are going to help the elongation factors and all these other factors kind of get recycled so they can come and do this again. But each of those is going to require energy because you're having to add a new GTP each time that you do this. So then you have those 3 triphosphates, kind of 4 per peptide added. Then you have those energy spending for the initiation phase as well as for recycling the ribosome. So it's an expensive process, but it's worth it. And all of this might just be the start of the process because once we have translation, we've got this quote unquote final protein, well, now we can do things like add post-translational modifications. These include things like phosphorylation. We can get ubiquitination, or some people say ubiquitinylation, and then there's all this debate, but you can get ubiquitin added. You can get hydroxylation, sulfation, nitrosylation, glycosylation, lipidation, all sorts of ations, all sorts of variations to post-translational modification. So these bells and whistles that we add after we already kind of finished making the protein, those bells and whistles that we can't code for directly in the genes for the protein, but we can add afterwards, and this can do things like change what the protein interacts with. It can regulate the protein's activity. Things like adding a phosphate group, well, you've got this big, bulky, negatively charged thing, and once it's like you've packed your suitcase and everything fit all nicely. And now you remember you forgot to pack your big spike ball. And so if you go pack your big spike ball, things are going to have to shift around. And this is going to cause a conformational change in your protein that may activate it. It may inactivate it. And even if it doesn't change activity, it might open up new binding opportunities or shut off binding opportunities if the binding partner doesn't want to hang out with the phosphate. So there are all sorts of ways that we can modify proteins after they're made post-translationally. But in order to do that, they've first got to get translated. And that's where we get our beautiful dance of translation. Sometimes I like to think about it as a series of sort of continuous weddings between amino acids, where our ribosome is kind of like this prefab 2-part chapel. Then our tRNAs are like limos that are bringing in our brides and our grooms. And they park in these kind of parking lots that are specified by the codons. And our elongation factors, these are like chaperone escorts that are going to be handling things. So basically we have our chapel with these 2 parts, the small subunit, the large subunit. The party starts by building that chapel around the first groom, which is brought in by a special limo or initiator tRNA. All the future ones brides and grooms are going to be brought in by the chaperone escorts, and which are our G proteins. So we have our F2 or our EF1. They're going to know where to park because they're going to have their parking spots specified by the mRNA's codons. And it's going to kind of match the limo's license plate. You're going to keep doing this, keep adding to that growing chain until it's time for the chapel to close. So the stop codon is going to enter the A spot. It's kind of like our decoy car. Our release factor comes in and it cuts off the chain. So if you want to think about things like that, that's totally cool by me. but also know the actual biochemistry going on behind the scenes.